Hello and good evening. So, how are you all doing? I hope everyone is in good health. So, how's your study going on? See, in the last class, we had discussed about Unit 11, and today I'm again back with Unit 3, Part A and Part B. By this time, I hope you have completed the study of Part A and Part B of Unit 3, that's Replication and Transcription, right? Today, in this series, we will mainly focus on questions which are a bit diagrammatic or tabular kind of. I do have questions which are in paragraphs for you as well, but mostly it is diagrammatic or paragraph based question, tabular based questions. So normally we did the part C questions. Okay. Today we'll specifically focus on these kinds of questions so that you get a very clear idea about how to analyze those diagrams, how to find out the tables and all. Though if you have a good concept about Unit 3, any kind of question should be easy for you to answer, whether it be a paragraph question, whether it be a diagrammatic question, or whether it be a tabular question, match the column kind of. So, this is Unit 3 once again, and are we all ready to test our knowledge? If you're all ready, let me see BIO in the chat box. Come on. So, are we all ready? Yes, good evening. Is my audio clear, everyone? Please, can you confirm? Yeah? Okay. All right. So, the first question for today's session is on your screen. Let's see if you can crack this question. And I'm starting with the very familiar paragraph kind of questions for you. Okay, multiple choice. You have to select more than one combinations. After this, I will get into the diagram and the tables. So this is a question from your post-transcriptional processing of RNA in eukaryotes. And as we know that eukaryotic RNA definitely has to undergo some post-transcriptional processing, and there are many kinds of processing that takes place, of which capping, polyadenylation, and splicing is the normal one. Apart from that, you have other specific processing also. So read the statements very carefully and select the correct combination of statements. So it is saying that following statements were made about the post-transcriptional processing of RNA in eukaryotes. Number one statement, they are saying soon after transcription initiation, RNA polymerase 2 pauses approximately 30 nucleotides downstream from the site of initiation until the cap structure is added to the 5' end of the nascent pre-mRNA. Statement number B, the 5' splice sites are functionally divergent, whereas the 3' splice sites are functionally equivalent. Statement C, in addition to helping in recognition of the splice sites, the exon definition also functions as a splicing regulator by allowing pairing and linking of adjacent 5' and 3' splice site. D, the intron definitions mechanism applies only to the larger introns above 500 nucleotides in length and assist in achieving alternate splicing. And number E, the splicing reaction carried out in vitro have revealed that the first and the second trans-esterification reactions are reversible. Okay, I'm getting some answers. Come on, I would expect all of you to give me some answers because this is the third time we are discussing Unit 3, Part A and Part B, isn't it? So, by this time, I hope everyone has revised these parts, have studied these parts. Come on, try it, everyone. This is all about testing your knowledge. Any more question? Any more answers? All right, so let's see the correct option, and the option is 4. Very good, all of you. Those who have answered, you have answered correctly. Good. Yes, yes, good. So let's see what they are saying. First statement, soon after transcription initiation, RNA polymerase 2 pauses 30 nucleotide downstream from the site of initiation until the cap structure is added to the 5' end of the nascent mRNA. We already know that your transcription initiation happens by two phosphorylation. Actually, initiation is done by one phosphorylation and then elongation phase takes place after the second phosphorylation. Right? So that is why the second phosphorylation that happens on serine 2, which we had discussed earlier also, that is a phosphorylation which marks the addition of cap structure because that recruits the capping machineries, the splicing machineries, both, right? So RNA polymerase initiates the transcription after the first phosphorylation and then it pauses until the second phosphorylation happens and the cap structure is added and then it again continues the elongation phase. So therefore statement A is correct. Statement B, they are saying the 5' splice sites are functionally divergent whereas the 3' splice sites are functionally equivalent. No, this is wrong. In addition to helping in recognition of the splice size, the exon definition also functions as a splicing regulator by allowing pairing and linking of adjacent 5' and 3' splice site, which is absolutely right. When we study about splicing, we get to see this process happening in that process, isn't it? The diagrams itself explains this particular statement, so this is correct. The intron definition mechanism applies only to the larger introns. No, introns are the non-coding sections. So whether it is a smaller one, less than 500 nucleotides or greater than 500 nucleotides doesn't matter. It is a non-coding sequences present within the coding regions, right? So therefore, this statement is wrong. D is wrong, and E is right. The splicing reaction carried out in vitro have revealed that the first and the second trans-esterification reactions are reversible. Right? So see, over here, Even if you don't know this particular these two statements, even if you don't know whether these two statements are right or wrong, okay, or even if you don't know this statement is right and wrong, you are confident about these two statements, right, A and C. That is no doubt about that. So from there itself you can answer the question. That is option 4 is correct. Okay. Okay, now going to the next question. So next question is a match the column question. So try to solve this. Read the question very carefully. A researcher wanted to test the effect of different chemical agents on double stranded DNA. dsDNA was taken in tubes A, B, C, and D, and four different agents were added individually to each tube. The researcher, however, forgot to label them. The properties of the added chemical agents on dsDNA were analyzed. The possible product, which is in column X, and the specific action of the chemical agents are listed in column Y. So you have to correctly match it. Okay. So if you find it to match from X to Y, you can go from Y to X if it is easy for you. So you have a double-stranded DNA. Think about it. You have a double-stranded DNA. Okay, and the composition you already know uh, how it is constructed. The double-stranded DNA. So we have the base, we have the sugar, we have the phosphate, sugar, base, right? This is how we get to see it. This is one strand, and over here we have the hydrogen bond. So it can be a double bond, it can be a triple bond. We have another base, we have a sugar, we have the phosphate, base, sugar, and phosphate, right? Base, sugar, and phosphate. So you have to remember the different kinds of bonds, the structure, and all. Okay, so many of you are giving me the answer. So let's see what's the answer. The option is option 2. The correct option is option 2. So therefore, if you want to get the nucleotides, that means what? You have to break the phosphodiester bonds, right? You have to break these bonds. So base sugar phosphate. Okay, base sugar phosphate. Base sugar phosphate. So if you want to get this here, then you obviously have to break the phosphodiester bonds which are formed. Next, only nitrogenous bases if you want to get, that means this bond you have to break, right? This bond you have to break. This is the glycosidic bond. So with this you have B with 4. Nucleosides, if you want to get, nucleoside means only the base and the sugar, right? So that means you have to remove the phosphate from here. So nucleosides means it is 2, C with 2. And single stranded DNA, if you want to get, that means you have to break all these hydrogen bonds. So therefore break hydrogen bonds. Very good, all of you. So you just see, very simple question, but if you get confused about what they are saying, you will not be able to solve these questions. Otherwise, the rest of it is very simple. Everyone is familiar with the general structure of DNA and the different bonds of the DNA, right? Moving to question number 3. So this is a, you know, diagram based question where they are saying the long DNA strand depicted below is serving as a template for lagging strand DNA synthesis. The short line represents the newly synthesized Okazaki fragments. At which position among A, B, C, D would DNA primase act next? Very interesting. Usually they say that which is the first fragment of Okazaki that has been synthesized. That is a very common question that is asked. The first Okazaki fragments, but this time they are saying that where would DNA primase act next? And we know that in order to synthesize a fragment of DNA, you have to first have the primase, otherwise, the DNA polymerase, will not be able to synthesize the stretch of deoxynucleotides. So, this is your lagging strand, right? Look at the polarity that will help you to analyze. So it is 5 to 3 prime, and replication or transcription always happens. Both replication transcription both happens from 5 prime to 3 prime. Right? This is the polarity of the synthesizing strand, the strand which is getting synthesized. So against that, the template strand will always be from 3 to 5. Think about it. So next, okay, some of you are giving me answer as C, answer as D. So let's check what is the answer. It is option B. Look at the positions they are saying. Obviously, primaries won't be sitting here. This is already a synthesized strand, right? The primer was already, it was here. So from there, the strand has been synthesized. The primer will not be sitting here because here you already have a primer. Okay, here you already have a primer over here, which has synthesized this particular strand B. Okay, and if I draw the polarity of it, then obviously this is 5 and this is 3. This is the growing end. This is 5, this is 3. Right, this is the growing end. 5, 3. This is the growing end. Okay, now here also there was a primer, because if there wasn't any primer, you wouldn't have synthesized this strand of Okazaki. So the next primer is supposed to sit here, against which you will be synthesizing the next Okazaki. Okay, so the correct answer is option D. Those who gave me the option as option C, I hope you have understood why it is not C, right? So use the concept of polarity, that will help you, and please observe the question very carefully where it is pointing. Okay, they are pointing at some positions. A cannot happen at all because this things has already it has been already synthesized, right? Yes. So see, this is a tricky question you have. Simple, but a slight misunderstanding, a slight observation error can get you the wrong answer. Never mind, practice it well. You will definitely be able to solve these kinds of questions. Okay, the more you practice, the more easy it becomes. And don't rush. Don't rush with these questions. Okay. Okay, again, a paragraph based question I have for you, the very familiar kind of question. So, this is again from your transcription. Total RNA was isolated separately from cytosol and nuclei of human cells growing in a cell culture. Each sample was mixed with a purified denatured fragment of a DNA corresponding to a large intron of a housekeeping gene, okay, and incubated under the renaturing condition. Given below are the statements made about the outcome of the experiment. So when you get these kinds of questions, no, just underline the important things. So you are taking the total RNA separately from the cytosol and the nuclei. So think about it. What kind of RNA you will get if you take the cytosol, if you take the RNA from the cytosol and if you take the RNA from the nuclei? Will it be both the same or will it be a bit different? Okay, this is the first question you need to ask yourself. The second thing that you need to do is you need to match this kind of information with what else they have given you. Like you're trying to mix this sample with a fragment of DNA corresponding to large intron of a housekeeping gene. Okay, under renaturing condition. Now, when you have analyzed this paragraph, now you go through the statements. Again, be very careful as to what they are telling you. So far, I've just got one answer from Suresh. Okay, Aruna has also given me an answer. I'm expecting answers from all of you because this is a very fundamental uh, concept, a very fundamental concept. While we study the difference between eukaryotic and prokaryotic transcription, we definitely talk about this. When we talk about the elongation phase of transcription, we talk about this. Okay. All right. So I've got a couple of answers. Let's see. The correct answer is option 3. It is option 3. Why? What is it saying? Let's see. In the first case, okay, first let us analyze the cytosol and nuclei, okay? Now, the RNA which is in the nuclei, so RNA transcription definitely takes place within the nucleus of the cell, isn't it? So, in the nucleus, this is a nascent RNA which needs to be modified. Remember, this is a eukaryotic one, okay? So, it needs to be modified. So, what kind of modification happens? First, there is capping, 5 prime cap. The second, you have splicing, right? And then you have the polyA tail. Okay, so therefore over here when the mRNA is present, okay, the RNA is present, it is likely that it will still have some introns, okay, because the process is going on, so it's likely that it will still have some intronic sections. But the moment it comes out to the cytosol, it is understood that it doesn't have any introns anymore, because you will not be transported to the cytosol until and unless the transcription is over and the entire processing has been completed, right? So there is no chance, even if over here we do have some chances of getting an intron, the intron which has yet to be spliced. And but over here when it is in the cytosol we will definitely not get any introns. Okay, so therefore statement number 1, RNA isolated from nuclei will form RNA DNA duplex because of the presence of introns in the primary RNA, which is right. There is a possible chance of it. Next, cytosolic RNA usually will not form RNA DNA duplexes. Why? Because by the time you're getting to the cytosol, obviously you have spliced out all the introns and you have the 5' cap and the poly A tail, everything. Okay, so A and B is correct. C, both cytosolic and nuclear RNA will not form RNA DNA duplexes. Okay, as transcription and splicing occur simultaneously. It is true that transcription and splicing occur simultaneously, but you are splicing out one intron at a time, right? So there is a chance that there will be some introns while you have extracted, the time you have extracted, there can be the presence of some introns, okay, which are, as I said, which are yet to be spliced, okay, because splicing is not complete in your nucleus. Okay, it's not complete until and unless you finish the entire transcription and even some time after the transcription. So therefore this is wrong. Okay, cytosolic RNA will form RNA DNA duplexes because unspliced cytosolic RNA are exceptionally stable. This is wrong. You don't get unspliced cytosolic RNAs. So I hope you have understood this, right? So moving to the next question. Again, this is a table based question. So given below are a set of enzymes in column A and enzyme activities in column B. Column A, all right, just a second. I will just go back. Surrender, in the question they are saying that in transcription splicing will occur simultaneously. No, it occurs simultaneously. Surrender, in case of eukaryotes, splicing and transcription and splicing will occur simultaneously. Eukaryotes, okay? Okay, for this question, the correct answer is option 1. All right, for option 1, so in this case, see, this is a very straightforward question. There is nothing to analyze here, okay? These are very subjective question. So you just have to know the properties of this enzymes and where they are working. That's it. Okay, very subjective questions they have asked. unlike the previous tabular question where there was a bit of analysis that you need to do. So straight away we have DNA polymerase delta. It is responsible for Okazaki fragment synthesis in eukaryotes. Polymerase epsilon, it is required for your leading strand synthesis. Okay. Topoisomerase 2 is required for double strand break and ligation, whereas topoisomerase 1 causes single strand breaks. So, very straightforward question, very simple question. See, right? So, study your enzymes very thoroughly. Okay, this is all about your enzymes. So, enzyme studies, the polarity. I just with these question answer sessions, I'm just trying to give you an idea of uh, about all the places from where the question can come. Okay, the different kinds of question that you can get. So while you're studying, you should focus on each and every sections of it. It is not if this time it has come from this particular section, the next time also it will be asked from that particular section. No. So if you notice, I showed you once that when they show the Okazaki fragment, the most common kind of pattern is to find out which is the first Okazaki, which is the last Okazaki. But in 1 year they have twisted the question and said that where the primer will be attached. Okay, the pattern is the same, but they have twisted the question slightly. So that is why this is the main reason why I'm again I have come back to unit 3, and this time I have focused on the diagrammatic questions and the tabular questions specifically. And Watson scholarship, you all know about it. Those who don't, this is for you. All right, so admissions are still open, and it is open for your Junoon batch. So you will get a lot of advantage here: 1 year coaching, 12 months of access to global and national workshops, our programming internship, 4 certification courses of your choice, direct access to your coaching experts on WhatsApp, and one VIP pass to our CRISPR conference, the CRISPR conference which will be taking place in 2023. So moving on to question number 6. Observe the diagram very carefully. You know what? Whenever you get to see a part C question, most of the students, like 80% of the students, they instantly become scared looking at the length of the question. Such a big paragraph. God knows what is written over there. Immediately they get the panic. But if you study the paragraph carefully and keep on marking the important things which are there, read it once, read it twice. For the first time, just read it and see what they are telling you. Second time, read it and mark the important things which you think is relevant to or required to answer that question. Okay, and then just string it together. You will see the paragraph, no matter how big it is very simple. And one such example is this question. It is looking like this is a very big paragraph, and many things are written over there. But it's very simple. Okay. Okay. Right. It is hindering the option. All right. Just give me a minute, please. Just give me 1 minute. I will just change the mode. Okay. I hope now it will be visible. I've shifted the options for you. Okay. So this is your one, this is option two, this is option three, this is option four. Okay, those who are giving me option as B, observe the diagram very carefully. See, they are telling you that this is a newly identified bacterium having a double-stranded circular DNA genome as it's shown. Okay, so characterization of DNA polymerase responsible for genome replication showed that DNA synthesis occurred in 5' to 3' direction. It depends on the presence of a primer. Polarities of DNA 3' or 5' are shown here. Replication begins at the point marked O, which is this point, okay, on the left of the bubble, and both the parent strands were replicated concurrently. Okay, that means same at the same time. The longer arrow inside the bubble shows the leading strand. So this is your leading strand. Okay, this is your leading strand, and these are your Okazaki fragments. Okay, these are your Okazaki fragments. So the model depicts what? See, if this is your point of origin, okay, on one side, the leading strand is going towards your right, the lagging strand is going towards your left. Okay, there is no bi-directional movement, there is a single direction. If it had been bi-direction, then you would have had the origin here, somewhere here, and on both the sides the replication would have happened. Okay, on both the sides, this is bi-directional, but in this case the origin is here and there is only one directional movement. Okay, so definitely the first two options are not valid, because in both the cases they are saying it is bi-directional. Bi-directional not at all is wrong. So you have to choose from option 3 and option 4. Now, in option 3, along with unidirectional mode of replication, they are saying that wherein the synthesis of the Okazaki fragment marked C occurs prior to those marked A and B. Okay, so here they are saying that C is occurring first and followed by B and A, whereas in option 4 they are saying that fragment A occurs prior to those marked B and C. So you basically are going to get confused between which is the first fragment. C is the first fragment or A is the first fragment. Okay? So see, again coming back to that same pattern. Which is the first fragment, but how they have twisted the question. Right? And you some of you got confused between the bidirectional mode. Bidirectional, unidirectional. The correct option is option 3. Why? Because if this is your origin, okay, and if this is the way your leading strand is moving, that means it's understood that your helicase was placed here. Now it is moving towards this side, because your leading strand moves in the direction of the movement of helicase. So whichever side your helicase is moving, your leading strand also moves towards that side, right? So that means the movement of helicase is from the left to the right. If this is what is happening, that means this will be the first Okazaki that would have been synthesized, because as the helicase is going this way, it is opening the gap, right? This part is open. C has been synthesized. Helicase is now here. It will open up this part. So then B will be synthesized. Helicase will move here. It will open up this part. Then A will be synthesized. Like this, it will go on. All right. So those who made a mistake, I hope you have understood why you went wrong. I hope it is clear. Now, moving to question number 7. Again, this is a very subjective question, but look at the pattern. They have given you a fill in the blanks kind of. Okay, and you have to, the answers are given in your answers only. X, Y, Z, it is given in your answers. So, this is your option 1, this is option 2, this is option 3, this is option 4. This is very subjective. You just have to know which polymerase transcribes what. Okay, I'm getting some answers. Okay, 3, 4. The confusion is between 3 and 4. Others? Only handful of you are responding. What about the others? Try it. Doesn't matter if you're going wrong. This is all about practicing. This is not the CSIR exam that you're giving. This is all about self check. Okay, and we are practicing it together. Try it out. Any other answers? Can I get some more answers? All right, fine. The correct answer is option 3. All right. Okay, good try. Good try. I know this is a bit confusing. I understand. But you have all tried it. So, that is a big thing. So, RNA polymerase 1, we have studied that it transcribes the rRNAs, isn't it? But look at the beauty of the question. Normally, what we study is that RNA polymerase 3 transcribes your 28S rRNA, the 5.3 rRNA, 5.8, sorry, and you have the 18S rRNA, right? We all normally study like this, that polymerase 1 transcribes these rRNAs. But what it is in total known as? In total, what it is known as? It is your 45 srRNA. So they haven't, yeah, 5.8 srRNA. So in total, this comes to as 45 srRNA. But this is something we usually don't refer to. So see, this is the beauty of the question. You already know, you already have studied. Okay, but here is where you have to link it up. So it is your 45. Okay, but I understand you are getting confused about 45 and 18s because you have studied in this manner, this format. Okay, but look at the next one, miRNA. This will clear your doubt. Even if you're confused between the X, the Y when you're trying to solve it, it will clear your doubt because miRNAs, they are transcribed by polymerase 2. Okay, not polymerase 5. They are transcribed by polymerase 2 and in some cases polymerase 3 as well. Okay? Now, even if you get confused by miRNA, what about the RNA polymerase found in plants? This is very specific. You have the polymerase 4 and polymerase 5, which are transcribed by the plant, okay, which are present in the plant, not transcribed. I mean, they are present exclusively in the plant. So there, the doubt will go. Okay, so this will be RNA polymerase 5. So this is all matching. Okay, this is wrong. This is wrong. Even if this is right, this can be right, but this is wrong. So automatically, if 3 is matching, then automatically option 3 is the right answer. Because miRNA, when you study about the miRNA, you will have to study about the polymerase too. Because miRNA, they have a specific gene from where the miRNA is produced, unlike the siRNA, right? So they are normally transcribed by your polymerase too. I hope it is clear. And siRNAs, these are your transverse siRNAs which were first discovered in the plants. Okay, they were first discovered in Arabidopsis, and there it was seen in the plant. It was seen that it is being transcribed by another special type of RNA polymerase, which is a polymerase 6. Yeah, exactly. Confused in miRNA and 18S. Okay, so see, if you know the miRNA, if you keep this in mind, then this will be sorted. Yeah, exactly. Okay, but good try, everyone. Good try. I highly appreciate your participation. Very good. Okay, question number 8. We are towards the end of the series because, like the other days, today also I have 10 questions for you. So, again, this is a paragraph based question where you have to select the correct statement. And it is a statement made by tRNAs. You see, most of the questions in CSIR, if you see, they are mainly concentrating on the mRNAs, the polymerase and all, okay, centered around that, or miRNA, siRNA and all. But yes, at times they do ask regarding your tRNA. So those who are thinking of skipping tRNA, okay, because tRNA processing you will be studying when you're going to study about the post-transcriptional processing. There are many things which are processed in the tRNA primary transcript. So those who have asked me the question in class that whether it is important or not, and now also you're thinking at the back of your mind that tRNA, whether it is important or not, here is a question of part C. You do get questions from tRNA as well. Okay? And TRNA, don't think about skipping it because it's a very small thing that you need to study about. Unlike the mRNA, where there's a lot of things that you need to study, TRNA is a very short paragraph. Because just a handful of processing happens here, and you don't even have to go into the very details of those processes. Okay, so they are saying that the following statements are made with reference to the fact that tRNA are known to possess T in their sequence. Okay, so RNA polymerase 3 utilizes TTP as one of the substrates. Like any other RNA polymerase 3 also utilizes RUTP. But when RNA pol 3 reaches a loop structure, it bends to S adenosyl methionine to methylate C5 position of the incorporated uracil, because thymine is your C5 methylated form of your uracil. Okay. During transcription of tRNA genes at the designated position, DNA polymerase replaces RNA polymerase 3 to incorporate T in the tRNA transcript. A specific methyl transferase utilizes a methyl group donor to post-transcriptionally modify the specific U residues into the T residues, and uracil to thymine conversion occurs in the large number of tRNAs in the T-C loop. The correct answer here is option 4. This is wrong. Okay, it doesn't utilizes TTP. No. Okay, this is also wrong. Utilizes UTP, but when the RNA pol 3 reaches a loop structure, it bends to this. No, it's wrong. Okay, the loop structure happens later on. You don't form the loop structure. The primary transcript is not looped. It loops after the processing happens. So during transcription, tRNA genes are designated at the designated position. DNA polymerase replaces RNA polymerase 3 is wrong. A specific methyl transferase utilizes methyl group donor to post-transcriptionally modify the specific U residues into T. This is the correct one. Okay. So when you're going to study about the D loop and the T side loop, you will have to study about these modifications, the diuridine modification and this methylation of the uridine that takes place. Okay, and the methylation of the uridine will give you the thymine. Okay, that is why it is T size C. And uracil to thymine conversion occurs in large number of tRNAs because tRNA structure itself, if you see, I'm drawing a very bad structure of tRNA, but yeah. So you have the CAA attached here, right? You have the D loop, you have the T psi loop, and here you have the anticodon loop. This is the general structure of your tRNA, isn't it? So option 4 is the correct one. Only D and E is correct. So next time, go into read the tRNAs. Okay, the processing is very simple, just few steps. That's it, not a very elaborate one. Okay, so quickly going to question number 9. Very interesting question. This is very interesting. There is a crosstalk between the DNA repair mechanisms. Okay, when you have a repair, it's not just fixed to one particular pathway. Yes, definitely, one particular pathway is very efficient in removing that problem, or repairing that problem. But if somehow that pathway gets mutated for some reason, if that pathway is not working, then will you sit with the mutation? No. The cell always have a plan B. If plan A is not working, there is plan B, and that is by going through the alternative repair mechanism. So you use a different pathway to rectify that repair. Okay, and this is a case that was observed in the colorectal cancer. This is a study that was found when the scientists, the researchers, they were studying the colorectal cancer. Yep, exactly. So the answer is option 2. Okay, you go by the alternate DNA repair pathway for repair, and most of the time it has been seen that it is a translocation kind of repair that is done. Okay, okay. This is the last question. We are to the end of it. It's of the series. So they have given you a construct was generated to access the activity of the bidirectional promoter. Remember, it's a bidirectional which has a common regulatory DNA element X. Okay, so this is a common regulatory element. You have a promoter which is working this side, a promoter which is working this side. Now, see, the activity of the promoter is recorded in the presence of increasing levels of E. coli encoded regulatory protein which binds to DNA element X. So, what you get to see if you analyze this graph, this is given to you. Okay, so if you analyze the graph, you will see that with increase in the level of the regulatory protein, what is happening? The basal transcription that was happening, it is increasing. Okay, for reporter gene 2, that means which is becoming more active, it is this promoter that is becoming more active. Okay, PB, this is upregulating this particular gene 2. But for your promoter A, okay, what is happening when the regulatory element is becoming more and more active, or increasing the level of the regulatory proteins, the basal transcription level is getting down. So that means you're suppressing it. Okay, you're down regulating the activity of this gene 1. So one promoter is upregulating the activity of one gene, the other promoter is down regulating the activity of the other gene, right? So when you are taking a mutant E. coli with mutation which abolishes the binding regulatory protein to eliminate X. That means X cannot bind. Then what kind of expression will you get to see? Yeah, I knew most of you will be giving me the option 2. Option 2 is not right. This is what I can tell you. Analyze the graph. What is happening? You have a basic level of transcription. This is the basal transcription. After that, it is getting modified with increase in the regulatory protein activity. Right? So when there is no regulatory protein, then what will you get? You will get the basal transcription only. So wherever you had, the gene 2, you will get the basal transcription of gene 2, gene 1, like that. Okay? No, not 3. Because look here. Look here. The basal transcription level itself is at this position. For gene 2, it is down. For gene 1, it is up at the top. Okay. So wherever it is, the basal transcription will be maintained only. Okay. Will be no other modification. Okay. So, with this, I come to the end of my session. I hope you found it interesting. Okay, meet you in another session with another topic. Thank you very much for attending. Come prepared for the next one. See you soon. Bye bye.